All right, we've given everybody 5 minutes to join. If they hop on during the webinar, we can answer any questions at the end. We're going to start off with some quick introductions, and then we're going to press play. I'm Rich Valentis. I am the general manager here at Catachem. I've been with Catachem for just about 2 years now. My background is mainly molecular biology. I've spent the last 15 years in CLIA, JCO-regulated labs. I've worked for some big players as a medical technologist, such as Yale, LabCorp Quest, Hartford Hospital, mostly in the state of Connecticut. Before joining Catachem, I was up at a couple private biotechs out in Boston, and this is my first foray into medical device manufacturing and lyophilization. It's been quite the learning curve for me, but wouldn't call myself an expert yet, but I've certainly learned a ton about lyophilization working here.
Yeah, good morning. I'll be giving the talk later on. My name's David Templeton. Background is primarily enzymes. I started my career with Genzyme Corporation, then joined Diagnostic Chemicals Limited, where we manufactured diagnostic reagents, dealt with raw materials, made some enzymes as well. I joined Catachem 15 years ago, again, working on diagnostic reagents, freeze drying a lot of proteins, primarily for calibrators and controls. So a pleasure to give you some thoughts on freeze drying this morning. We prepared the talk a few days ago, so please excuse a secondary introduction once the slideshow begins.
Just some quick information about Catachem before we start. We've been in business since 1984, so just over 40 years now, mainly in the veterinary market for chemistry reagents. Catachem has been doing lyophilization internally for the past 15, 20 years and have expanded our capabilities to take on customer projects and CDMO activities for Lyo in the past 4 or 5 years. That said, we're going to switch our camera off to save some bandwidth and start our webinar. Thank you very much for joining.
Instant coffee is lyophilized coffee, which is reconstituted with water before drinking. Lyophilization, the name, comes from the Greek word lyophile, which means likes water. The process is simple. Water freezes into ice, think snow on the ground. When it remains cold, this snow, rather than melting into water, sublimates, i.e. changes directly into vapor without melting. Skipping the liquid water phase is the essence of lyophilization. The product that remains after lyophilization, the instant coffee for example, is now dry without having experienced extreme stresses during the drying process. Commercial lyophilization speeds up the sublimation process using pressure differentials, i.e. vacuum, as well as temperature differentials in the apparatus used. We call this machine a lyophilizer or freeze dryer, which is represented by this illustration on the slide. At Catachem Incorporated, we have 4 of these machines, all manufactured in New York State by Millrock Corporation. These are all designed to assist in scale-up from an R&D phase to commercial production. The information obtained from our smaller R&D system is directly translatable to recipes for use on our larger equipment. Again, if the process is simple, then what is all the fuss about? A good question that we hope that this webinar might help to answer. Lyophilization is an expensive, energy-draining process with many variables to control. The primary one being heat transfer, stretched over many hours. The key is to optimize the process to save resources and manufacture a consistent, cost-effective product that conforms to all required specifications. Before going any further, it will be useful to provide a couple of explanations to those unfamiliar with the lyophilization vernacular. A recipe is both a formulation or mixture of components designed to assist the freeze-drying process. Additional buffers, sugars, proteins, and other components are often needed to enhance the process, as many materials do not freeze-dry well on their own. These additional components are called excipients, cryoprotectants or bulking agents. The second part of a freeze-dry recipe is the instrument program, outlining the temperature controls, the vacuum or pressure controls, and the timing involved in the process. For the scientists listening, if you've ever tried to take a freeze-drying cycle that works beautifully for a handful of 2 ml vials and scale it into hundreds or thousands of vials or into multi-liter trays, you already know the reality. Scaling lyophilization is not copy-paste. It is translation. The physics changes, the risk profile changes, and the controls you need to maintain consistency get more demanding, not less. Today I'll do 3 things. First, I'll walk through the full lyophilization process end to end, what actually matters in each phase, and the key parameters you have to design around. Second, I'll connect those fundamentals to scale up. Why trays behave differently than vials, why 100 ml vials behave differently than 2 ml vials, and what practical engineering decisions keep quality consistent. Third, I'll explain how CATACHEM operationalizes these, how we move from R&D to pilot to production while maintaining quality, documentation, and reproducibility under an ISO 13485 quality system. We'll close with the science of product shelf life extension, why lyophilization stabilizes sensitive reagents and biologics, and what failure modes it prevents. Here's the agenda that you'll see today. What lyophilization is and why it's still the gold standard. The process overview, freezing, primary drying, secondary drying, and closure. Key development parameters and how we determine them. Scaling formats, vials versus trays versus beads, and the physics behind the differences. Operationalizing scale, from R&D batches to production runs while keeping quality intact. Shelf life extension, the science and what it enables for shipping, storage, and performance. And then briefly, some practical, frequently asked questions: moisture, uniformity, closure integrity, oxygen management. Let's start with the why. Lyophilization or freeze drying remains the benchmark for sensitive materials because it removes water gently. It avoids the high temperatures and shear forces associated with alternative drying methods. For many proteins, the enzymes, antibodies, nucleic acids, and diagnostic reagents, that matters because the product's value is its function after reconstitution. In a regulated world, it's also a predictable, documentable process. A successful cycle is not just a dried cake, it's a validated thermal and mass transfer pathway that can be repeated and defended in audits, in tech transfers, and in long-term stability data. At CATACHEM, most projects start with one question: what do you need the product to do after reconstitution? Items like activity, potency, calibration accuracy, reconstitution time, and stability at defined storage conditions are all important considerations. Then we engineer the cycle and the format around that. In one sentence, lyophilization is freeze the product, reduce pressure, sublimate ice during primary drying, then desorb bound water during secondary drying, all while staying below critical product temperatures. Lastly, seal the container to preserve what you've achieved. This sentence sounds simple. The complexity is that every variable interacts: formulation, fill volume, container geometry, shelf mapping, freezing rate, chamber pressure, ramp rates, and the endpoint definition for moisture. If you change one, you often change 2 or 3 others, with the results as varied as those shown in the vials that are illustrated. Phase 1 is freezing, and in many ways it determines everything that follows. Freezing creates the ice structure, and the ice structure becomes your pore network after sublimation. Larger, more open ice crystals generally produce larger pores, and less resistance to vapor flow, which can shorten primary drying and improve uniformity. But freezing too slowly may create phase separation or destabilize some active ingredients. Freezing too fast can create very fine ice crystals, a reduction in pore size with less space for water vapor to escape, and therefore slower drying. So what really matters in freezing? Freezing rate and nucleation. And what is nucleation? As ice forms, the water molecules cluster to make nuclei, or seeds, from which larger ice crystals grow. The more controlled this is, the less haphazardly the ice crystals form, and the more uniform the process and the final freeze-dried product. Final freeze temperature and hold time is important. Whether the formulation benefits from annealing is also important. So what is annealing? Annealing is the process in which a gentle heat step is added after the initial freeze to regulate ice crystal size before refreezing. At Catachem, we treat freezing as a design step, not a checkbox. As a solution freezes, the unfrozen liquid concentrates. The pH can change dramatically. Cryoprotectants can separate from proteins, so the same proteins can freeze under different conditions. A good freeze cycle is designed to protect the weakest protein and prevent osmotic shock. For vials, we can achieve uniform shelf freezing and map behavior with product probes. For trays, we focus on fill depth and thermal gradients. And for sensitive formulations, we evaluate whether controlled nucleation or annealing improves outcomes. When making freeze-dried beads or lyo beads, the reagent droplets are flash frozen in nitrogen, so control of freezing here is impossible. However, other formulation variables are required to maintain the integrity of the lyo bead. These beads, for those not familiar with them, are tiny freeze-dried spheres of product, 10 to 20 microliters in volume, that freeze dry as spherical nuggets. We will talk more about these a little later on. Primary drying is where most water is removed, and it is also where most failures occur. In primary drying, ice sublimates under vacuum. The shelf provides heat. The chamber pressure and the condenser pull vapor away. The product temperature must stay below the collapse temperature for amorphous systems or below the eutectic melting point for crystalline systems. These terms will be explained in more detail on the next slide. This is where people can make two common mistakes. First, they monitor chamber conditions and assume the product follows. It does not always follow. Second, they push shelf temperature for speed without truly knowing the product's thermal limits. At Catachem, primary drying is controlled around product temperature, not just shelf temperature. We use product probes and cycle analytics to ensure the product stays within its safe window. The goal is not as hot as possible. The goal is as warm as allowed safely, because that's how you balance throughput with protection. Collapse temperature, TC, and eutectic temperature, TEUT. Awareness of these temperatures for a freeze-dried product are the key determinants in designing a successful freeze-dried procedure. The eutectic temperature is the melting point of a crystalline product as it freeze-dries. The collapse temperature for an amorphous product is where it starts to soften and lose structural integrity. There's also the glass transition temperature, TG, where an amorphous material shifts from a hard, brittle glass to a softer, rubbery state. Although close, the collapse temperature, where the freeze-dried cake actually caves in, is usually a few degrees higher than the glass transition temperature. Collapse is not just cosmetic. Cake collapse is a physical signal that the product matrix exceeded its structural limit during drying. That can translate to reduced pore structure, slower reconstitution, higher residual moisture variability, potential loss of activity or potency over time. A product that looks dry can still be compromised if it crossed collapse limits even briefly. So the process question becomes, how do we define and respect those limits? We build the primary drying steps and pressure targets around these limits, and as scale increases, we recharacterize, because on scaling, the thermal pathway changes. Secondary drying removes bound water, the water that didn't leave as sublimated ice, but is attached at the molecular level to the formulation matrix. This phase can be the difference between a product stable for 12 months and a product stable for 24 months or more. It is also the phase where the tendency and easy route is to run a generic secondary dry phase of a basic recipe. This can be dangerous, and it is where value can quietly leak out. In secondary drying, you typically increase shelf temperature under continued vacuum to drive desorption. But you must remain below thermal limits for the active ingredient, the excipients, and the container and closure system. Overheating can denature proteins. Under drying can leave residual moisture that is too high, leading to stability failures months later. At Catachem, we treat secondary drying as an engineered endpoint. We define a moisture target range tied to performance and stability, and we confirm it with quantitative methods, typically Karl Fischer titration. We do not rely on time alone as proof of dryness. Residual moisture is one of the strongest predictors of long-term stability. Too high, and you accelerate hydrolysis, oxidation, and other degradation pathways. Too low, and some proteins can become overly brittle or lose activity depending on the system. The point is not lowest moisture possible. The point is validated moisture range that correlates with retained function and shelf life. Practically, that means defining the moisture specification and sampling plan, testing edge and center positions to confirm uniformity, trending moisture across runs for process repeatability. This is one of the places ISO 13485 discipline directly supports product performance. It forces consistent execution, consistent sampling, and consistent recording. Stoppering, sealing, and the last 2% of risk. After the cycle, you still have one more reality. The product is now highly hygroscopic and vulnerable. We mentioned the Greek word lyophile earlier, likes water. The moment you expose it to ambient air, it can absorb moisture quickly. Closure integrity and atmosphere management become decisive. At this point, you have choices. Stopper under vacuum, backfill with nitrogen or other inert gas, or do both. Maintain vacuum during stoppering and then apply an inert gas backfill strategy, depending on the product needs and the equipment configuration. The decision should be driven by oxygen sensitivity and closure integrity considerations. Now let's address a common misconception that a vial is a vial. A 2 ml vial behaves fundamentally differently than a 100 ml vial because geometry changes, heat and mass transfer. Small vials have high surface to volume ratio, efficient heat transfer through the glass, and shorter vapor pathways. Large vials have deeper product beds, higher resistance to vapor flow, and greater temperature gradients across the frozen matrix. If you run a 2 ml cycle on a 100 ml vial without redesign, you invite incomplete primary drying in the lower region, hot spots near the surface as you compensate with temperature, non-uniform residual moisture, and collapse or melt back risks during transients. So scaling by container size requires re-engineering, shallower fills, adjusted shelf steps, and a pressure strategy aligned with the increased load. This slide shows the different energy profiles, i.e. heat and pressure, that relate to successful lyophilization as vials get larger. Vials versus trays, and why scale-up is really an exercise in controlling heat and mass transfer. So let's move from vial scaling to tray scaling, because this is where many projects stall. In vials, much of the product has direct thermal contact through the glass and shelf, and the geometry is consistent vial to vial. In trays, depth and surface area become dominant. Fill depth increases resistance to sublimation. Edge effects can be pronounced. Thermal gradients across the tray can create zones where product temperature and drying rate diverge. Scaling is not same recipe, just bigger. It is engineering a pathway that recreates the same product conditions, especially product temperature relative to collapse temperature across a different geometry. When scaling to trays, three practical controls dominate. First, control fill depth. Shallow fills freeze and dry more evenly. Deeper fills increase resistance and create a higher probability of incomplete drying and local temperature gradients. Second, match freezing with protocol to scale. A controlled shelf freeze at bulk scale helps prevent non-uniform crystal formation. In trays, non-uniform ice can create non-uniform pore structures that then translate into non-uniform drying. Third, monitor product temperature, not only chamber conditions. You need probes in multiple locations, center, edge, potentially multiple depths depending on the format, to verify the product never drifts above collapse temperature. At Catacam, we operationalize this with test batches and temperature mapping in representative loads. It is a faster path to a robust recipe than guessing with time alone. Vials versus bulk trays versus lyo beads. A strategic decision that should be made early is format. Traditional vial lyophilization is ideal when the end user needs ready-to-use containers with robust stability data and minimal handling. Bulk tray lyophilization is efficient for intermediates, for materials destined for downstream subdivision, or where high throughput drying is needed before final fill. Bead lyophilization is well suited for automation, unit dosing, and workflows that benefit from free-flowing formats, especially for PCR and other high throughput assays. Just a few additional comments about lyo beads. As mentioned before, the lyo bead is a spherical bead formed when a small drop of a liquid material, 10 to 20 microliters, is dropped into liquid nitrogen where it quickly freezes. The bead is then collected and freeze-dried in the second stage of the process. A good bead is uniform, has structural integrity, is easy to handle, dispense, and package. One of Catacam's first lyo beads, containing the protein bacteriorhodopsin for use in human retina replacement therapy, was sent to the International Space Station for experiments in manufacturing under near zero gravity. Manufacturing stable lyo beads is a unique discipline, but not one we have time to elaborate on today. Suffice to say, choosing the correct format early prevents costly redesign later. A common failure mode is developing in vials and later realizing the commercial workflow demands a tray or bead format. This change in design can add months to a project. So what makes a custom recipe truly custom? A freeze-dry recipe is not a list of shelf temperatures. It is an engineered relationship between formulation thermal limits, ice morphology and pore structure, product temperature behavior under load, chamber pressure control for stable mass transfer, and endpoints that correlate with performance, moisture, reconstitution, potency, and stability. Two formulations with similar concentrations can require very different cycles because excipients, buffer salts, protein structure, and fill geometry change the collapse limit and the resistance to sublimation. Catechem's value here is not that we run a cycle. It is that we can develop the right cycle quickly, prove it with data, scale this to commercial volume, and then lock the process into controlled manufacturing. In practice, we track 5 variables as the backbone of process control: product temperature, chamber pressure stability, residual moisture, cake morphology, and reconstitution performance. These are not academic. They are the variables that predict whether your stability data will hold up, whether your end users will get consistent results, and whether your lots will remain commutable across time and geography. Here's how we typically structure development. First, we define target product performance and constraints, storage conditions, required shelf life, reconstitution time, potency or activity, allowable excipients, and regulatory pathway. Research use only versus in vitro diagnostics or chemical intermediate expectations. Second, we characterize formulation behavior. What bulking agents and stabilizers are appropriate, whether crystallization is desired, and what thermal limits govern the cycle. Third, we run iterative R&D cycles with product probes and cycle analytics. The purpose here to define a safe and efficient primary drying pathway and a validated secondary drying endpoint. Fourth, we apply robustness testing. Small variations in fill volume, load configuration, and parameter tolerances to confirm that the cycle is not fragile. A cycle that only works under perfect conditions is not a production cycle. Research use only and in vitro diagnostics use differ in the level of documentation and validation expected. For research use only, teams often prioritize speed and flexibility. You still need stability, but you can move faster and iterate more freely. For in vitro diagnostics or chemical intermediate production, consistency and defensibility become dominant. You need validated specifications, controlled documentation, traceability, and evidence that the scaled cycle reproduces performance, lot over lot. Even container closure selection becomes part of the validation story. Catechem operates under ISO 13485, as we have mentioned before, which supports in vitro diagnostic expectations and provides structure for its projects, where customers want disciplined manufacturing and documentation. Shifting from science to operations. In research and development, you may run dozens to hundreds of vials. In production, you may run thousands of vials, multiple shelves, multiple loads, and more complex staging. The equipment can differ as well. Operational scale introduces higher thermal mass and longer equilibration times, increased risk of shelf-to-shelf variability, greater need for validated loading patterns, increased importance of standard work, filling accuracy, loading consistency, stop ring parameters, and post-process handling. The answer is not run longer. The answer is controlled scale-up, mapping, validation, and disciplined execution. One of the least visible but most critical safeguards is shelf mapping and chamber characterization. Even advanced freeze dryers can have small temperature gradients across shelves and positions. A few degrees can translate into meaningful differences in product temperature and drying rate, especially near collapse limits. This slide shows a 5-degree change in temperature across one shelf. We address this by mapping thermal distribution, designing load configurations that minimize edge effects, using representative probe placement to validate real product behavior, and updating recipes to account for system-specific performance. This is a key reason a cycle developed on one system cannot be assumed to transfer perfectly to another without additional translation work. At production scale, quality is maintained through controlled documentation and execution, controlled recipes with defined ranges, training and qualification for operators, in-process monitoring and deviation handling, defined QC release testing, moisture, appearance, reconstitution, potency where applicable, and lot traceability and change control. The theme is consistency. Customers do not just need one good lot, they need the fifth lot to match the first, and they need the documentation to support that expectation. Shelf life extension requires considerable development and often redevelopment. Too much water in a product drives degradation. Lyophilization slows these degradation pathways, but that perfect balance can be difficult to find. Addressing shelf life extension directly, proteins are structurally complicated, as this slide illustrates. Add water and complications increase. Most degradation mechanisms in biologics and diagnostic reagents depend on water. Hydrolysis requires water. Many mobility-driven reactions accelerate with higher water content. Microbial growth requires water. Ice crystal melting and refreezing cycles in liquids can damage proteins during shipping. By removing water, lyophilization reduces the rate of these pathways dramatically. It does not eliminate all degradation, oxidation, and other pathways can still occur, but it shifts the system into a much more stable state. In practical terms, a properly designed lyophilized product can maintain activity and calibration integrity longer, tolerate broader shipping conditions, reduce reliance on cold chain, and reduce waste and lot failure risk for the end user. Proteins are fragile because their function depends on precise structure. During lyophilization, if shelf temperature rises above the collapse or denaturation threshold, proteins can unfold or aggregate. If you remove water too aggressively without stabilizers, you can disrupt the hydration shell and the matrix that protects conformation. Gentle water removal means product temperature control during the primary drying, a stabilizing excipient system to protect the structure, secondary drying that achieves target moisture without thermal damage. The output is not just dryness, it is functionality after reconstitution that is the key metric. Oxygen, moisture, and closure integrity. Even a perfect cycle can be undermined by packaging failures. If closure integrity is compromised, even microscopically, oxygen and moisture ingress can occur over months. That can change residual moisture, accelerate oxidation, and undermine stability. So we treat container closure selection as part of development, selecting vials and stoppers appropriate for the product and storage duration, ensuring crimping and stoppering are controlled, implementing inspection and, where appropriate, integrity testing strategies. This is one of the quiet variables that separates lab success from commercial reliability. Lyophilization is not just for one material class. At Catachem, we support a range, including enzymes and other proteins, antibodies and other biologics, nucleic acids and molecular reagents. Also complex diagnostic matrices, including controls and calibrators, as well as emerging modalities where structure preservation is critical. The technical requirement changes with each. Proteins may be collapse limited, nucleic acids may be pH and excipient sensitive, and complex particles require careful control of freezing and drying to preserve functionality. A generic recipe rarely works. The freeze-dry cycle must be built around the molecule and the intended use. In diagnostics, for example, controls and calibrators must perform consistently over time. A calibrator in a diagnostic test provides the yardstick from which all results are ultimately derived. There is no room for error or variation in its manufacturing process. In the slide, the results shown by the blue dots here should be as close to the red dashed line as possible. Lyophilization improves this by slowing chemical degradation pathways, reducing microbial risk, enabling more stable shipping and storage, improving lot-to-lot consistency when moisture and morphology are controlled. When your business depends on reproducibility, lyophilization becomes not just a stability choice, but a quality system choice. Let's define zero compromise in real terms. It means that when you move from 2 ml vials to 100 ml vials, or from vials to trays, or from hundreds of vials to thousands, product temperature stays within validated limits. Residual moisture stays within a validated range. Reconstitution and potency stay consistent. Cake morphology remains uniform and acceptable. Documentation and traceability remain complete. Achieving that requires both science and operations. You need engineering to translate heat and mass transfer across scales, and you need a quality system to lock the process down. Let me finish with some answers to common questions that we often get. For example, can we just run the same recipe longer for larger volumes? Longer time helps only if the underlying heat and mass transfer conditions are appropriate. Scaling requires re-engineering, shelf steps, pressure strategy, fill depth, and probe validation. There are no shortcuts. A question may be, what's the single most important variable in long-term stability? Well, the answer would be residual moisture in combination with closure integrity. Moisture is the driver of many degradation pathways, and closure integrity determines whether moisture stays controlled. Another question, how do we know a primary drying cycle is complete? Well, you confirm that by monitoring the process signals on sophisticated equipment, the equipment that you're using. Complete drying is not just a time point, it's a validated endpoint. We have questions about vacuum or nitrogen backfills, the difference. Well, driven by oxygen sensitivity and container closure performance, primarily. Many sensitive products benefit from aggressive oxygen reduction with closure integrity considerations guiding the final approach. What about vials versus trays versus beads? Well, you start from the end user workflow and you scale backwards. Format selection early prevents expensive redesign later. So putting on my sales hat for a minute, what would Catachem provide as a lyophilization partner? Well, it would provide cycle and formulation development grounded in measurable product constraints. It would provide scalable translation across vials, larger fills, and bulk formats. In-process monitoring focused on product temperature and controlled endpoints. Moisture validation and QC release testing aligned to stability needs. Operational discipline under ISO 13485 for consistent auditable production. If your goal is to go from proof of concept to reliable supply without surprises, our focus is to remove uncertainty in the cycle and remove variability in execution. So let me close with this. Lyophilization is often described as freeze it and dry it. In reality, it is one of the most precise manufacturing processes in science. Scaling it successfully is not about bigger equipment. It is about controlling the product's thermal and mass transfer environment across changing geometries and loads. When you do it correctly, you get extended shelf life, reduced cold chain burden, and improved global shippability. coupled with consistent product performance and, where applicable, unwavering regulatory compliance, the result is less wasted energy and a better product. Thank you so much for joining me today. I'm happy to take any questions that anyone might have. Thank you.
Okay, all. Thank you very much for attending. If there are any questions, please drop them in the Q&A, the chat, and we can answer them live or afterwards. You're very welcome. We're going to work on getting the recording out in the next couple of days. Okay, well, if nobody has any questions, then we can go ahead and, oh, you're very welcome. Nobody has any real questions, we'll feel free to drop off. I see your question, Barry. So as far as secondary drying is concerned and the endpoint for that determining, we have Carl Fisher titration to establish what the residual moisture is. We also can verify it gravimetrically based on loss on drying. A combination of those two with the reconstitution and the cake morphology are usually pretty good indicators of what the residual moisture is and how the product will perform later on. Hopefully that answers your question. David, anything to add to that?
No, that's the way we do it.
All right. So we don't currently have mass specs on site to analyze the finished product. We can partner with some outside laboratories if that is a requirement of a particular project, but right now it's not part of our capabilities.
Well, we will soon be introducing a HPLC system that we will be able to use, but not quite mass spec as yet.
Mhm. All right, everyone. Appreciate your time today. Thank you very much for joining. And there's last chance for questions. If not, we'll go ahead and give everybody 7 or 8 minutes back. And thank you for joining.
Appreciate it. Thank you.
Ready. Take care, all.